So our next speaker is Tatyana Sharpee, who is getting wired up there, and who was an early collaborator of Allison's. Thank you.
Thank you very much. I appreciate the opportunity to speak to you today. It's a great honor, and I thought that today I wanted to discuss. I had the honor of working with Allison, and we published one paper together. Actually, it was supposed to be more than one paper, but it's kind of glued together into one piece that was inseparable. We had a theory and experiment, and following on the themes that Bill Bialik mentioned, that Allison was discussing with him and others at the Keck Center. My project with Allison was to think about adaptation in the songbirds, and basically what we found that any possible element in our model that could adapt would adapt. And my job was to figure out and quantify how exactly that would happen. You heard a lot about Allison's interest in neuroethology and learning, but she was also had an interest in auditory processing and the basic mechanisms of auditory processing. For these songbirds are a great system because they can perceive sound so well. So in Allison's lab, my project was to analyze how neurons in an analog of the auditory cortex, area L field L, respond to stimuli. And one might notice that this area is multiple synapses removed from the periphery. So this poses somewhat of a computational challenge of how we relate the stimuli to the neural responses that are measured. And some of the technique Frederick Tennyson mentioned, and I will go into more detail along those lines. So the underlying framework that we wanted to work with is how are we going to connect the stimuli that are present, the sounds that are presented to the bird, to the spikes in a given area. And I often think of these experiments as what happens if you get a present and it's all wrapped, and you would like to know what's inside without opening it. So we shake it. So you see how it responds. So this is kind of the same idea. We have a brain, and there is a neuron. It's all wrapped. So we present some stimuli, and it's random stimuli, but at some point we observe responses. And what happens is that by chance, some of the fluctuations approach closer to what neuron is searching or responding in the stimulus. And these slight fluctuations in the firing rate we can pick up through averaging statistics. So in some ways, to me, statistics is like magic, because if you take a very long sequence of data, and it's the same data, and you present to different neurons, after averaging, you will find that they're selected for different features, because after averaging, everything that was not related to the spiking of that particular neuron will go away, and everything else will remain. So the elements of the model of how we characterize neurons are trying to understand which aspect of the stimulus triggered the response, and we model them as linear features or filters. And then because neurons are inherently nonlinear, we also model them, include a nonlinear component in the model which relates the spike rate to the output of this comparison characteristic. And as you can see from the list of references, which is not exhaustive, this approach has been very successful in a variety of sensory areas. including originally it was pioneered by Doupe and Cooper in the auditory system. So that was what was available. But then, of course, the reality is that neurons are selective not just for one feature of the stimulus, they're selective for conjunctions. We want to see what are the events that are happening in the outside world, and those are characterized by conjunctions. So in order to explain this, the model needs to be made more complicated. So instead of saying that each neuron is picking up only one set of feature in this stimulus, maybe it's paying attention to two or more. And now the prediction for the producing the neural response, the neural firing rate, becomes a two-dimensional function, which is depicted here as color, meaning that darker colors would denote high responses, meaning that the conjunctions when this feature is present strongly and that feature is both present strongly, then the neuron will produce the response. So one can build a hierarchy of models to understand how that the signals are processed from the periphery through the sequence of stages of transformation. And in that context, I started to work with Allison, especially because most, so far, I've been talking in the context of these stimuli that, as I said, like shaking for the gift. But as Frederick and Bill Bialek mentioned, there is this emphasis for understanding how neurons operate in a natural environment, or how the auditory neurons process songs. So following advice from Bill Bialek, we asked the question of whether it's possible to estimate these features using not noise stimuli, but natural sound stimuli. And one can, so one could argue or ask why would one ever use noise stimuli if you can use song stimuli? But the problem is that once we have structured input, it's very hard to analyze it, because if I have something structured and as the result of the analysis I get a structured output, I don't know whether that structure is due to the input or was the selection due to the neuron. So that's the beauty of information theory that was suggested by Bill Bialek is to find the statistical technique that would pick out of the stimulus those features that are relevant by finding which features carry most of the information in a technical sense, as in bits, about connecting the stimuli and the neural response. And I was curious, so after developing this algorithm in model neurons, I was curious about applying this to real data. And I talked to Allison, and first we applied it to visual stimuli with Mike Stryker and Ken Miller. And then, but what was inspiring to me in the auditory data set is this graph. I will tell you a little bit of personal story that is associated with this. So this is one example of a neuron in the auditory field analog of auditory cortex. And this was one of the first neurons in a data set that was collected by Kathy Nagel, where through the analysis, I picked up that these neuron responses were based on two features, something which looked like an integrator, and the other one which was sensitive to the derivative. And it looks simple now, and we have now applied it to many other algorithms. But at the time, it was the first time when I ran the algorithm, and we saw a clear second feature in the case of real neural data. And that's when I I was impressed that my own code worked. So at the time, I was a little, for some reason in my mind, it made a connection with flying. So I'm a theorist by training. And before that project, I didn't have the large experience with writing numerical methods. So I was a bit apprehensive whether my algorithm can find all these minima and overcome the numerical optimization. And then I'm sitting in the airplane and I'm thinking, well, I remember this calculation in physics textbook about how the airplane flies and the lift force. And the calculation is based for an infinite wing. And I'm looking outside in this little airplane, and the wing is not infinite. So this somehow that made a connection that even though the algorithm may not be perfect, it still works in the real situation. So then we analyzed it for many neurons, and we found that there were these two features, and they had different patterns for each neurons. And what was interesting is that when the sound intensity would change, each of the neurons would alter the patterns that it was sensitive to. And there was a systematic pattern, which I will only summarize in the abstract form, where in the space of kind of three dominant ways that the features could take, the first feature would change in one systematic way, and the second feature would change in the other, meaning that neurons adapt to sound intensities. But not just features changed. Remember that in the model there are elements which describe what are the features that neuron is picking out in the neural response. But there are also nonlinear part of the model which says how does these features affect the neural response. And it turns out that these functions were all complicated. And at the time, we could say we observed the diversity of these nonlinear functions, and they would change depending on the variance in the loudness that Bill mentioned in his talk. So that left me with a question of what is how to characterize them, and how much time do I have?
You have some time.
Okay. So that left me with the question of how to characterize these shapes, because at the time we published this paper, I could only say that there is a diversity of shape, but I didn't know what would be the best way to describe these nonlinearities. And here we were inspired again by ideas from Bill Bialik's group, but also going back to Jane's theory, who asked the question of, given a certain data and measurements, what should be the best, how should we describe it? And so for example, if we have some distribution of stimuli that have the same mean and variance, all of these distributions that I have drawn, they all have the same mean and variance. So if we measured mean and variance, I can, which of these distributions should I choose? They're all consistent with the data. But somehow our inner instinct says I should choose a Gaussian distribution. That's a second nature. So James quantified that contribution by saying that's the distribution that is maximally uncommittal, but respects the measurements of the data. And it's true, maybe the data is described by more other distributions, but until we have evidence for it, we shouldn't impose it on the data. And there is actually, right. So we want to generalize this idea, but not just for the distribution of neural responses, but for distributions of relating stimuli to neural responses. In other ways, going back to this picture, we wanted to find what are the minimal functions that would describe this complexity, consistent with certain measurements that we made in the lab. So it turns out that there is an answer for that. And the answer is a function that many of us are familiar with. It's a logistic function. And interestingly, it's in many biological systems, including transcription and in neural responses. And the equation is, suppose I have a set of points like this, and I can fit it with some relationship. So one possibility is to have a threshold linear function. It has two parameters, a threshold and a linear slope, so that's reasonable. But it turns out that it would make some assumptions about high order moments, about how this the value on the x-axis is related to the y-axis. So it turns out that the logistic function does not make these assumptions, and it's a direct analog of the Gaussian distribution. So here is my one equation. We can ignore it, but basically that the minimal model for neural responses is a logistic function whose argument can be either a linear function of the input, which would correspond to this traditional model of the receptive field, where the neural response is a function of the strengths of stimulus component along this axis. But it could also be expanded and to take into account, so the quadratic term here, the second order, just meant to represent, one can think of it as conjunctions of features. So once we take into account these high order interactions, all of this model, this type of model, can produce a diversity of nonlinear functions. And now we have a framework for modeling a diversity of neural responses that are observed in auditory, but also in other sensory systems. So analyzing, following up on this, we then expanded. And for example, this is actually a matrix. One can diagonalize it. One can analyze more than two features, many numbers of features. And one can also make sense of these features, and in some case, put them in the right combinations in order to infer the features of neurons that feed into the neuron we are recording. So in this example, one analysis would produce what's called an orthogonal representation, which is just an artifact of the method. But then we can invert this to find the features that are localized, and in this case, it's in the retina, and therefore provide a way to infer putative receptive fields of inputs. So I would like to summarize the research part of my talk by saying that inspired by Allison, we studied the adaptive properties of auditory neurons, that features change, nonlinearities change with respect to variance, and we can now develop a set of techniques that make it possible to estimate more than two features, estimate different types of nonlinearities, and in some cases reconstruct the circuit, what happens before the neuron that we are recording for. And in summary, as a way of thanking Allison for everything that I owe her in my scientific and personal life, I wanted to highlight one aspect that hasn't been mentioned yet. So Bill mentioned succinctness, but I also missed her humor. And also being non-native speaker, I admired her precise use of words. And I just wanted to share one short email that I received from Allison when I submitted a paper for publication. So Allison said, "Fingers crossed, egos prepared." So I think that's one of many. Another quote that I really like is when I was saying that I can't really bring food for the lab meeting, I have something else. And she said, "Tanya, you always need to try to do more. So, and I think it characterizes, she always tried to do more. Thank you for your attention.